Our third speaker is Dr. Sriram Sankararaman. And Sriram is a professor in computer science, computational medicine, and human genetics here at UCLA. I have sat in Sriram's talks several times. I just want to tell you, every time I learn something new, which also sparked some of my students' dissertation work. So today, I definitely look forward to learning again. Yeah, let's welcome Sriram.
Thank you. Thanks for the invitation. It's a great honor to be talking to you all today. So today I'm going to say a little bit about our work, which has been really inspired by what we call biobank-scale data. So essentially this kind of relatively new phenomenon of these large repositories connecting genetics and very diverse phenotypes, and what are the kind of statistical computational challenges that it entails for us. But before doing this, I also wanted to spend just a few moments talking about Ken's influence on my work and the way I view science. So I was, when I was starting my position here at UCLA, I was planning to teach my first graduate course. It was a course on machine learning in bioinformatics. So the goal was to get students from computer science, machine learning students interested in this field. And I was searching for inspiration. It wasn't clear, how do you teach a course like this? And one of the books that I came across was Ken's fantastic books, amongst many of his books, on methods for genetic analyses. And it's a beautifully written book, very lucid, but also what came away was kind of this guiding principle, which I think of as the blessing and the curse of interdisciplinarity, right? So interdisciplinary research, you get to work on some really interesting problems, important problems, but the challenge is you do require investment of learning this additional field. So there is no getting away from that fact, right? Whichever direction you come from the biological direction, you have to learn statistics and computation. When you come from statistics and computation, you have to learn something about the biology. So this was, of course, this is Ken's words that you can read here. And the other one is, how do you convince people who have no background in biology to learn biology? Because it seems like a mess, no real rules. It's just like a series of random facts. And again, this is Ken's words, you can read here. This idea that although it seems like, I mean, at a face value that there is nothing here, there are underlying principles, there are underlying rules, and that it is worth spending the time to learn it as much as possible. Okay, so I should also mention, Ken, in this preface, he says, and this is a real thing, that writing books often seems like a daunting task, something you would rather spend time writing your next grant or paper. But I should say, yeah, all of Ken's books have been a huge source of inspiration and instruction. Okay, so now with this, I'd like to talk about biobanks. So biobanks are essentially what I said, these large repositories connecting genetic data with lots of diverse phenotypes. They've been around for a while, but what has happened over the last 5-10 years is there's been a real kind of push in increasing the scale and the diversity of these data sets. So we have biobanks here at UCLA. We have biobanks in other academic centers in the US, in Europe, in Japan, and China. And for example, there is this famous biobank, the UK biobank, that's been quite prominent out of UK. And so as the scale of these data sets increases, one of the questions for me is the size of these biobanks is now in the hundreds of thousands, potentially millions. And many of these biobanks are getting to these sizes. So how can we leverage these large biobanks, millions of genomes, to gain insights into what we call genetic architecture of complex traits? We'll see what we mean by that in just a minute. So this I think of as a very interesting, challenging problem for us. And there are lots of different ways of thinking about it. There are questions of privacy and data access. These data sets are hard to access easily for groups that aren't connected in the right way. Of course, this is sensitive data, so how do you work with this in the right, with the right kind of protections? Interpretability, we've heard several people talk about it. When you have these complicated data sets, it's not even clear what interpretability really means. largely in the eyes of the beholder, but how do we embed those notions into our inferences? But for today's talk, I'm going to just focus on these two aspects. The first one is statistical issues that come across from these types of data sets. And essentially, the idea is we'd like to build statistical models that are as realistic as biologically plausible, given these data sets. But then there's always a tension. When you make your models statistically realistic, statistically complex, you run into computational issues, right? How do you apply these models? How do you do inference on them at this scale? So there is this inherent tension, this trade-off between statistics and computation, which I think is fascinating and something I'm very interested in. So now let's make this more concrete, and let's talk about a few types of questions that arise. The data that we'll be dealing with looks a little bit like this, for those of you who might not be familiar. So we have data on the left, which we call genotype data. Every row is an individual. Every column is a place in the genome where there is variation across individuals. The particular types of variation we care about are places where some people have one copy of an allele and other people have another copy. Technical name for these places are what we call SNPs. So the genotype data is going to be rows of SNPs for each individual. And then that is paired with a phenotype. Phenotype could be quantitative, binary like disease status, high dimensional phenotypes like imaging. But for now, we can think of it as a simple measurement like this. Now, what are the questions we might be interested in? Again, we can think of this. Oh, and because of the nature of human genomes, because our genomes are diploid, we have two copies of the genome, we can essentially summarize this data as the number of mutations that each person carries at a particular SNP. So depending on whether a person carries 0, 1, or 2 copies of an allele, you essentially end up with this matrix, this ternary matrix of genotypes across individuals. So the key kind of inference problems that we often encounter, and the only reason I'm listing these are the kinds of problems that we have thought fairly hard about. I'm sure there are others that are missing. The first set of problems focuses on looking at this matrix of genotypes and trying to figure out what is the relationships amongst the individuals in this matrix. So we heard from Matthew about ways of taking these matrices and understanding structure. Oftentimes, when you do this, the key kinds of components, the key kinds of parts that you derive are related to the populations these individuals come from. So we often call this population structure inference. The second set of inference problems are trying to connect genotype to phenotype. And we call this genetic architecture inference. So we'll spend some time on this. And the final one is focused on complications that arise when we look at the phenotypes. And this has to do with the fact that oftentimes there is huge amounts of missing data, and that needs to be dealt with appropriately. So I won't say too much about population structure, except that these issues of population structure are often posed as some type of matrix factorization. So you're starting with your genotype matrix, and you're trying to factorize it as a product of low-rank matrices with certain assumptions imposed on them. So depending on the types of assumptions, you might end up with models like PCA or admixture or topic models, right? Now, the challenge is how do you perform or compute these matrix factorizations when you have a data set where the number of individuals, number of rows is hundreds of thousands, number of columns is in the millions. So that's a computationally challenging task. And we've done some work on this. I won't spend too much time, but the key kind of insights here is our view of these as latent variable models that leads to inference algorithms that are sufficiently computationally efficient. And then we combine it with different insights from recent developments in linear algebra, randomized linear algebra, to get efficient inference. So this is just like a very brief overview. It's described more in these papers from our group. But now I'll move on to genetic architecture of complex traits. So what do we mean by this? So we have a trait or a phenotype, and this phenotype is a function of an individual's genotype and their environment. So broadly, we want to figure out what is this function that goes from genotype and environment to a phenotype. Again, that's pretty abstract. So now let's make it even more concrete. So we have to write down a model. So the simplest model is a linear additive model, where we write down the phenotype as a linear combination of an individual's genotypes, plus everything else, which is noise, sometimes also called the environment. So this is a fairly simple model. We don't necessarily think this is the correct model, but it's a good starting point, and we'll already see this leads to interesting challenges. So our goal is to infer this model. So that means, naively, we'd like to infer the effects associated with the SNPs in this model. So we'd like to make inferences about these coefficients. As posed, this problem cannot be solved because it's a high-dimensional problem. So you have many more SNPs than the number of individuals. So even with biobank-scale data, you have way more SNPs than the number of samples. So you need to put some assumptions on the effects of these individual SNPs. So common set of assumptions are, we assume that every SNP has an effect size that is drawn from some distribution. And the distribution, say, has mean 0 and some variance parameter associated with it. So we'll call this variance parameter the genetic variance component. And then you have the environment coming from a distribution with mean 0 and a variance parameter, which we call the environmental variance component. So essentially, we are integrating out the effect sizes of the SNPs. And now we have a model with these two hyperparameters, which is the genetic and the environmental variance component. Putting these parameters together, we get an important quantity in genetics, which is called the heritability. So the heritability is this ratio of the genetic to the total variance. It's a number between 0 and 1, and broadly, traits which have heritability close to 1, you can think of as highly predictive from genetics under this model. Close to 0 are relatively less genetic. So for those of you who don't work in genetics, you work in other types of data, other molecular data, say other non-biological data. Again, these concepts show up again and again. You can think of heritability as a measure of signal to noise. You have inputs and outputs, covariates and responses. You're trying to figure out what is the measure of the predictive signal in your features relevant to the output, right? So that's another way of thinking about this notion of heritability. It's a very simple model. Clearly doesn't seem biologically very plausible, but there are ways of extending this model. So one approach is, it seems unlikely that every SNP, no matter where you look at, is going to come from the same variance component. We know that there are different parts of the genomes. They have different potential impacts on function and phenotype. So this model could be adapted. For example, we might say SNPs that fall within protein coding genes, they come from one variance component. SNPs that come from somewhere outside of these genes come from a different variance component, right? So now you have two variance components related to genetics. And now this can be generalized. You can have lots of variance components depending on where these SNPs fall across the genome. So even these simple models can be extended, made more biologically plausible and realistic. And there's been quite a bit of influential work using these types of models to try to understand where the genetic signal in a complex trait is concentrated. So here's an example of one such study from a few years back. They were essentially applying this type of a model. And what they're plotting on the y-axis is a measure of enrichment of genetic signal in specific regions of the genome. So for example, you'll see that these regions of the genome, like regions which are evolutionarily conserved, tend to have high genetic signal for a variety of different traits. So this is the kinds of inferences that we would like to make from this model. So broadly, we have a variance components model. So you have your phenotype. We have taken our genotype and broken it up according to functional annotations, genes, other types of information. For the genotypes in a given functional annotation, we have effects associated with them. These effects are all drawn from a single distribution, parameterized by this variance component. And so this is a model where the parameters of interest are k such variance components and the environmental variance component. So this is what we'd like to be able to estimate from data. So we know how to solve this problem in principle, write down the likelihood, maximize, right? Challenge is performing this kind of a maximization scales cubically in the number of samples that we have. So n is the number of samples. So imagine biobank-scale data, hundreds of thousands, millions. Essentially, you have to perform an iterative optimization where each iteration is going to scale like this. So this kind of approach doesn't scale readily to large data sets. And there's been a lot of very creative algorithmic work in trying to drive down its computational cost. Still, there is a challenge in applying these methods to the kinds of data sets that we are encountering. So how do we proceed? So let's back up and look at alternate ways of performing estimation. So clearly, there are these two broad classes from basic statistics of doing estimation. Maximum likelihood is one. The other one is method of moments, another classical approach for estimation. And in fact, this method of moments has been used in the genetics literature. It goes under the name of Haseman-Elston regression, HE regression for short. So in method of moments, you write down moments, so sample moments, and you figure out values of the parameters so that the model's theoretical moments match sample moments. That's the basic idea. The way these models are defined, turns out there is no information in the first moment. It's always zero. So we look at the second moment. So the second moment, the covariance of our phenotype, can be written as this function of the parameters of the model. So you have all of the variance component parameters, and you have these matrices. So K is a matrix. It goes by the name of a genetic relationship matrix, sometimes called a kinship matrix. It's an n by n matrix, where every entry tells you what is the relationship between every pair of individuals at the set of SNPs corresponding to this annotation. So that's the model moment, and we have the sample moment. So we'd like to figure out values of the parameters such that these two quantities get close. So that's a very simple problem. The nice thing about method of moments is there's a closed form solution. So it turns out the method of moment solution is obtained by solving this linear system. So you can just solve it, provided you have all of the coefficients for this linear system. So that looks like progress, but we are not done yet, because we still need to compute these matrices, these genetic relationship matrices. So as I said, a genetic relationship matrix is a matrix that's computing all pairwise inner products between the individuals at a set of annotations that we are interested in. So it's an n by n matrix. So computing this will have n squared entries, quadratic in your sample size, and it has a pretty big constant, which is the number of SNPs that we are interested in. So still not there in terms of Biobank-scale. But now, if you look at this, it seems like there's a lot of wasteful computation going on. So you have a linear system here. What we need to compute the solution to this linear system are its coefficients. The coefficients of this linear system look like this. So they're taking our GRM, our genetic relationship matrices, and you only need to compute traces of the GRM or traces of products of the GRM. So that's the information that you need to get a solution to this linear system. So you're forming these huge matrices and then collapsing it down to a small number of trace computations. So that seems wasteful. So this leads us to an alternate approach, which is let's not form this GRM and then compute the trace. Maybe we can approximate the trace directly. This is a well-studied problem in, again, randomized linear algebra, which is the problem of stochastic trace approximation. This goes back to papers from the late 80s by Hutchinson. And the idea is if you want to compute the trace of a matrix, you can approximate it by essentially multiplying this matrix by a random vector on the right and the left. And as long as this random vector has this simple property, mean 0, covariance 1, this is an unbiased estimator of the trace. So do it once, and that gives you an unbiased estimator. Do it a few times, average it, and that allows you to drive down the variance of the estimate. The nice thing is you can now compute these traces without ever forming the GRM. So there's some additional work to show this. But all you need to do is multiply your original genotype matrix with some number of random vectors. So that's a new design parameter that we dealt with. For those of you who come from computer science, there's another way of understanding this. So essentially what you're doing is you're taking your large genotype matrix, multiplying it by a small number of random vectors. So we're working with a sketch or a random projection of the genotype matrix, which hopefully retains a lot of the information that we care about. So we are multiplying our genotype matrix with these random vectors. Clearly, the smaller the value of these random vectors, the number b, the more efficient, the smaller our sketch, and the more efficient our resulting method is going to be. So the computation scales like this. And if you're curious, I can tell you why you have these log factors. It turns out there is additional structure in the problem that can be exploited. So the efficiency is clear, but what is less obvious is what about the statistical efficiency or the accuracy of this method? And what is interesting, and I should say surprising, is these kinds of methods work, they're accurate for relatively small values of the number of random vectors B. So B could be as small as 10, and these methods give you accurate results. So we'll see this in a bit. So just recall your number of, your original matrix N could be 100,000 million by a million or 10 million. And by multiplying it with 10 random vectors, you've sufficiently reduced the size of your problem. There's another interesting aspect of this resulting algorithm, which is also desirable. It's what we call a streaming method, which means you never have to load all of your large genotype matrix into memory, which also makes it sufficiently memory efficient for use. Okay, so let's first look at some simple simulations. Gold standard is maximum likelihood. So how does this do compared to the gold standard? We can't evaluate it on the large scale data set. So these are small enough where you could do MLE and these randomized method of moments. We also call it randomized Huysman instant regression. So it's quite comparable. As expected, the standard errors are a little bit bigger, about 10% larger than maximum likelihood. But here is the main win, and that is in terms of scalability. So as you increase the sample size, so you're looking at sample sizes moving beyond tens of thousands to hundreds of thousands, these are some of the state-of-the-art maximum likelihood computations. And this is randomized Huysman instant regression. So this method is substantially more scalable as the sample size grows. It turns out there are additional benefits to this method. And as I said, we often don't think that the genome has just one variance component. There are different parts of the genome that have different variance components. We might want to fit each of them together. And this kind of a method allows you to fit many of these variance components jointly. So you can fit hundreds of variance components on, say, millions of SNPs and millions of individuals jointly. So these are the kinds of things which are much harder to do on maximum likelihood-based estimation approaches. So the upshot is you have a scalable method. By allowing us to fit these many variance components, you're essentially fitting much more expressive, much more plausible models at a fraction of the computational cost. So then we have applied this to data from the UK Biobank. Essentially, we are applying it to look at how much heritability different traits have as we look at more and more SNPs. So the number of SNPs is growing. So that's always this race that we're running where you have more SNPs, the size of the data keeps growing, methods have to catch up. And so as we increase the number of SNPs going from relatively common SNPs to additional SNPs that are substantially less frequent in the population, we can recover heritability for different traits. How much heritability we recover depends on the trait we are analyzing. For traits like height and BMI, it can be quite substantial. We can also ask where this heritability is localized across the genome. So essentially, we take these traits and we ask, is there an enrichment of heritability in different parts of the genome? Regions which are evolutionarily conserved have this enrichment that we observe across all traits. This is work that has been observed in prior studies. We also find that there are certain traits where there is enrichment that is trait-specific for specific regions of the genome. So again, enhancer regions tend to have enrichments that are very disease and trait-specific. So when I started looking at this, I said the reason why we want to start with linear models is, A, that was what was being used in the field, but we already have computational difficulties with fitting linear models. So now we have a method which can fit these linear models at scale. So this got us thinking, can we now move beyond fitting simple linear models? Can we increase the expressive nature of the models that are now being fit? And of course, can we estimate these models in Biobank-scale data? And what is interesting is this approach of randomized method of moments is general enough that it can be used in a variety of settings. So it allows us not just to fit the models that we just talked about, but models that I'll talk about next. So what these additional set of models do is allow us to explore types of nonlinear interactions which were not possible to explore previously. So I won't tell you much about the validation. The idea is these methods do work well in those settings. I'll just talk about the model and what we tend to observe after being able to fit those models on these large data sets. So the first type of nonlinearity in genetics is the following. So let's say you're looking at a phenotype and you're looking at how this phenotype changes as a function of the genotype at one SNP. So you have these three genotypes, and if it varies along this line, that's an additive effect. And if there is deviations, it's often called a dominance deviation. So it's kind of the simplest way of thinking of nonlinearity. So a question, and this is a debate in genetics, is there evidence for dominance deviation for complex traits? We see this for certain types of traits, but we haven't really been able to see this in the traits that are being measured on these data sets. So how do you fit this? So now you take your model, you have your additive component as before, and now you have a new component which quantifies these dominance deviation effects. And so now you have variance components for both the additive and the dominance. So in this model, we are jointly estimating the additive and the dominance deviation effects. And how we do this? Randomized method of moments. So essentially, we have kind of machinery which allows us to fit all of these without explicitly forming all of the large matrices using the same ideas that was discussed previously. So now we apply these models, these estimators, to data from the UK Biobank for about 50 traits, and we ask how much additive variance or additive heritability do we see across these traits, and how does that compare to dominance heritability? And the key observation is for traits, additive effects are substantial. This has been well known. So depending on which trait you look at, we have about 20 to 30% additive heritability across all of these traits. When you look at dominance, the picture is different. And you can't see maybe the axis on the bottom, but essentially it's consistent with 0. So across all of the traits we look at, there appears to be no statistically significant evidence for dominance deviation effects. And we can also do some power computations to see how small these effects were that we couldn't detect them on these large data sets. So now let's move on to the next source of nonlinearity, and this arises from context. So let's say you have a trait like BMI, and you're asking what is the impact of a particular SNP on BMI. So if you have a setting where the impact of the genotype at the SNP on BMI is modulated by an environment, say whether somebody smokes or not, then that would be an interaction between the environment or the context and genetics. So we call this G by E for gene-environment interactions. So now a question is there evidence for gene-environment interactions that we can infer from these data sets? What is nice about these biobank-scale data sets is often they don't just compute or they don't just collect one phenotype. They're often collecting lots of different phenotypes. So things like smoking, physical activity, lifestyle, diet, and so on. So this allows us to ask questions about different gene-environment interactions. So the way to set this up is the same basic principle. So we have our model, we have the additive component, and now we have this component, which is the G by E component. So we have an environment. We take the product of the environment with all of the SNPs in the genome. So essentially, we are asking in aggregate, is there an impact of the combination of the environment with all of the SNPs in the genome without necessarily pinpointing particular SNPs that might be contributing? So is there a signal from all of the SNPs in conjunction with this environment? And the effect is now quantified by the variance component associated with these effects. So we have a G by E variance component associated with these effects. Again, when you start forming these additional products, you have to be even more careful about computation and memory. So there is some technical work, but the bottom line is we can fit these models using the same principles mentioned. And we've been now applying these models to different combinations of phenotypes, environments. So for example, we've looked at sex as an environment, smoking status as an environment, medication usage, in this case, statin as an environment. And what we find is that there is evidence for different amounts of G by E, depending on the combinations of environments and phenotypes. For example, an obvious thing that shows up as a positive control, we have serum testosterone levels. And so there is substantial G by sex for serum testosterone levels. BMI has substantial G by smoking. This is well known in the literature. And then for statins, G by statin, there is substantial G by statin for things like cholesterol, but also other types of blood lipid measures. So these are things that we are digging into, but the key kind of takeaway from this is there is evidence for G by E contributing differently for different traits depending on the environment. So now the final part, the final type of nonlinearity would be interactions not with environment, but with other genetic variants. So this is what we call G by G. So the only difference is now, instead of looking at an environment interacting with the rest of the genome, we are looking at one particular variant interacting with the variants in the rest of the genome. And we're asking, essentially, is the effect of a particular variant on a phenotype modulated by all the other variants in the background of this individual? So this is a model that was introduced in this paper by Crawford and colleagues. And it's a particular type of gene-gene interaction. They call it marginal epistasis. So it's a very clever idea. The problem with typical approaches for finding genetic interactions is you would have to search for every possible pair of SNPs that might contribute to a trait. And that hurts your power a lot because of the multiple testing burdens involved. So here, what you're doing is you're not looking for a pair of SNPs. You're asking, is one SNP having its effect modulated by the rest of the genetic background? So you're still asking a hypothesis about one particular SNP. And so you don't pay the cost of a quadratic number of hypotheses. So you still hope to have some power to be able to detect these. So now the test is whether this marginal epistasis variance component is non-zero. And again, we can detect this using the techniques we just mentioned. So we can fit these models in UK Biobank type datasets. And what we find is when we apply these tests to several different phenotypes, there is statistically significant evidence for these types of marginal epistatic contributions from specific SNPs. So this is quite interesting because there's been a lot of debate about the contribution of interactions, the contribution of epistatic effects for complex traits. It's been very difficult to show this in human genetic studies. And so the ability to actually test and measure these in these large data sets could be quite interesting. We can also quantify the magnitude of these effects. So if you were to look at the magnitude of the effect explained by one of these variants compared to the magnitude of the effects explained by the variant in conjunction with its interactions, it can actually be quite a bit bigger. So this is telling us that these interactions can actually play a fairly significant role in how this variant impacts the trait. So this is all ongoing work that we are still trying to dig into and dive deeper into. Okay, I don't know how I'm doing in terms of time. You're fine. I'm good. Okay, so with this, I'm going to switch gears now. I'll talk about a different problem that arises when we decide to look at these types of biobank data sets. And this is focused on the phenotype side of things. Right? So far, I've been kind of droning away on scale and how that's good. But this picture is actually a little bit more sobering. Right? So you think, oh, you have all of these hundreds of thousands of individuals. Let's go and look at my favorite trait in this data set. And when you finally make the kind of computation, you'll find that the numbers are a lot smaller than you expect. And of course, the reason for this is many of the traits that are interesting, clinically relevant, are hard to measure, hard to collect in these large sample sizes. So essentially, for logistical reasons, cost reasons, a lot of these traits are only measured on a handful of the individuals in these biobanks. So an example is looking at how much percentage of a trait is missing across, say, about 300 traits that we tabulated. And you'll see that the missingness proportion is quite substantial for a lot of the traits, including traits like depression, which our collaborators are interested in. So how do you deal with this? The classical approach is to impute missing data. So the idea is you try to figure out a way to fill in missing values using the fact that there might be other measurements that have been collected. So the phenotype imputation problem is the following. So you use the fact that phenotypes 1, 3, and 4 might be correlated with phenotype 2 to impute the missing values in phenotype 2. And of course, what makes this challenging is there might be missingness in each of these other phenotypes. And so ideally, you'd like to jointly impute all of them together. Right? So that's the phenotype imputation problem. So this is a very well-studied problem. there's lots of methods trying to tackle phenotype imputation. However, there are still some criteria that we need from phenotype imputation methods that we felt were missing in these kinds of settings. The first one is we'd like these methods to be expressive. So we should be able to not just encode linear relationships amongst phenotypes, but potentially nonlinear relationships. Second one is they need to be scalable to be applicable to these large data sets. And the final point, which is an important one, is the missingness that you encounter in these data sets has structure to it that needs to be leveraged. So just to give you an example of what we mean by structured missingness, here's an example from this data set where there are these questionnaires that are being asked. So you have one questionnaire, and depending on the answer to this question, you have follow-up questions that get asked. So if a person answers or does not answer the first question, the follow-up questions are also not administered. So you have missingness that is determined by missingness in other entries. This is one type of structured missingness, but there could be other potential ways of having structured missingness. So how do we leverage structured missingness to do better imputation of phenotypes? If you knew something about the mechanisms of missingness, this is something that we could use in our model. In the absence of mechanisms, we will use an empirical approach. So we will use missingness patterns seen in our data to essentially train our model. So we call this approach copy masking. The idea is pretty simple. So let's say you have data sets where phenotype 2 and 4 tend to co-occur in terms of missingness patterns. So this pattern occurs very frequently. So then we would want our model to be particularly induced to predict missingness jointly across these two phenotypes. So we would simulate or induce or mask these two phenotypes much more frequently and train our model to perform imputation in that setting. So this is a type of inductive bias that we are imposing on this model. So that's the model of missingness. Now you have a model of imputation. You could use any model, but because we want something that's nonlinear, we are using an autoencoder. So this is an autoencoder framework where you're taking your original data, mapping it to some hidden layer, potentially lower dimensional, and then mapping it back to the original space. With that, you are reconstructing the missing entries. So you have an autoencoder which is trained on data where copy masking induces structured missingness, and that leads to this phenotype imputation method. So how does this method perform? We compare it to a few different alternatives. So soft impute is a method that came up in Matthew's talk. So soft impute is essentially a linear model. It's essentially a variant of PCA. We also looked at deep learning-based methods, which don't leverage structured missingness. And we also compared to classical approaches for doing missing data imputation. And so here we have measures of accuracy across different phenotypic data sets that we collected in the UK Biobank. And what we find is autocomplete across the range of missingnesses that were simulated is more accurate than these other methods. We can also look at individual phenotypes and see which are places where autocomplete is doing better than methods like soft impute. So with this, we then imputed phenotypes that were clinically relevant. So these were phenotypes that our collaborators were interested in. In this case, this was measures of lifetime depression. Turns out it's hard to accurately assess lifetime depression on these large biobanks. So it is observed in about 20% of the original data set. And now we imputed lifetime MDD in the full UK Biobank. And as a consequence, this larger sample size allows you to do certain downstream tasks with greater power. So for example, if you wanted to discover genetic risk factors for lifetime MDD, this depends on your sample size. So in your original observed lifetime MDD, if you ran a genome-wide association study, because of the sample size, we would find one significant locus. But now with imputed lifetime MDD, the larger sample sizes lead to more genetic discoveries. A worry in all of this is, could the imputation be introducing biases in the imputed phenotypes? So clearly, you might be finding things, but are these likely to be real and related to lifetime MDD? So we did some additional follow-ups in terms of trying to replicate these discoveries in other cohorts to see whether we are seeing consistent effects. we also tried to see whether the effect sizes are concordant across these different studies. And the general takeaway seems to be that the discovered genetic factors are replicable. Of course, there is no guarantee that in every such imputation task, that there is not going to be any bias. So how do you ensure this in a given imputation task? That is an ongoing thing that we are thinking about, but we don't have a good solution at this stage. So just to wrap up, so what I've taken away from this are, on the one hand, there are some interesting, I think, statistical and computational insights. We don't understand them yet, but I think they are interesting and worth pursuing. The first one is the effectiveness of random projections to summarize and compress data. So it's interesting that it works. What we don't have yet is an understanding of when and why it works. So a related question is, how do you design random projections with nice properties? So the simplest one is you just choose Gaussian IID random vectors. That's just one choice. But there are other choices that could give us different trade-offs in terms of computation and statistical efficiency. So that is something that we haven't explored yet, but I think it would be really interesting in these kinds of problems. The other one is the idea of leveraging structure in the data. I didn't say much about this, but it turns out essentially everything that we have described under the hood, you can think of as taking your genotype matrix and performing matrix vector multiplications. And because this matrix, genotype matrix, is structured, it has entries of zeros, ones, and twos, you can do this operation much quicker than if you ignore that structure. So again, there are some interesting insights into how you would accomplish those kinds of speed ups. And the other aspect of structure is when you're modeling missingness. So this is something we talked about in the context of phenotype imputation. In terms of the insights into biology, from the variance components work, I think the key takeaway is the substantial additive heritability, contribution of additive effects for complex traits, negligible dominance. And I think based on our preliminary work, I think there is emerging evidence for the role of interactions, both interactions with environment and with other genetic factors. And from the phenotype imputation work, I think this is a promising approach to combine hard-to-measure phenotypes collected on small data sets and scale them up to these larger biobanks. Of course, the question is how to do it without introducing biases and how to quantify the uncertainty in the process. These are things that we still need to work out. With this, I'd like to thank the students who led the work. So the work on the variance components was led by Ariel, Ali, and Boyang. And the work on missing data imputation was led by Ulzi. I think some of them might be here today. And with this, I'd be happy to take any questions. Thank you.
So how are you ensuring that causal predictors are included or incorporated in imputation? Like, because it seems like as we get more and more data, it seems like causal structures become less prioritized, and then it's like, how are we incorporating these?
Yeah, so given the way these data sets are collected, so these data sets are primarily observational data. In a sense, we aren't paying attention to the underlying causal mechanisms that give rise to them. But there is some way in which you can incorporate causal knowledge. And this is where I kind of briefly talked about how when you think of the mechanisms of how missingness arises, when there is causal information about how missingness arises. For example, in questionnaire data, you might have causal information about how missingness arises. So those could be the kinds of settings where you could inject causal knowledge to improve or deal with structured missingness. So yes, I think there is a need for combining our models and domain knowledge with these kinds of approaches. So far, we haven't done it because we've essentially been working with phenotypes that were just collected without paying close attention to how they were collected. So I think you have to go back to study design and data collection at that stage. But that's a great question.
Also, like thinking about, you're looking at an outcome to like depression, and you said it's only collected about 22%. It seemed like it would probably be more feasible to start with those who, it's still a large biobank, right? So then being only having to impute potentially smaller numbers in that subset of folks who have versus imputing 80%, which induce a lot of bias, especially in depression, coming from like a genetic framework, and you're thinking that this is a subset of the population. So I'm wondering, like, is there a reason to try to scale it to the whole biobank unless you started already with a smaller set, like maybe a subset of the 22% who have reported depression?
Right. So yeah, the question of, so these are individuals who have been measured for gold standard depression, right? So there isn't, or there could be reasons to try to impute and see if the imputation changes what was originally observed. But ultimately, you would want to move beyond the subset of 20% who were collected for gold standard depression. Whether you want to do it on the whole biobank or not, yeah, that's an interesting question. And again, we essentially, this was a thought experiment. Like, let's see what happens if we could impute into the whole biobank. Are you still able to get hits, and are those replicable? So that is kind of a question we're still trying to figure out as to what are the settings where this would work well, what are the settings that you can trust these imputed phenotypes.
The 20% was the people who were asked about it, not the people who were diagnosed with it. Right?
Correct.
So actually only a small fraction of those would actually have depression.
Correct. Yes.
So 20% sounds like quite a lot.
Yeah, these are 20% who were assessed, and of those, a subset would be diagnosed as MDD cases. So that's the interpretation of the 20%. Yeah, the 80% were not asked. They didn't answer the relevant questionnaires, which would have allowed an assessment of depression to happen. That's what's being imputed. Yeah.
I was wondering about the uncertainty for outcomes, whether there is a way already there is your software on the surface, and how that is incorporating downstream.
Yes. So yeah, the way we incorporate uncertainty. So this is following kind of classical multiple imputation approaches. Is essentially there is a bootstrapping process in which we are bootstrapping over the missing data mechanism, the model training, the initialization. So all of that uncertainty is being bootstrapped over to create multiple imputed data sets. And the GWAS, the results that I showed, integrates over those uncertainties. So once you have the multiple imputed data sets. And yes, it does increase the standard errors somewhat. So how much it increases depends on what's the fraction of observed to missing data. Yes.
So during the training process, like the training network, and then you were artificially like masking the data.
That's right. Yes.
Yes. So you need to have like a complete data, right?
You don't need complete data. Essentially, you're introducing additional masked or additional kind of simulated missing data using this copy masking technique. So these are entries where you know the truth and you've introduced missingness. So this allows you to compare the imputed outcomes with the originally observed outcomes. And that's where learning happens.
So we use like a subset data set, hopefully like more complete to train the model, and we apply more portion where it's one.
You don't need to restrict that. I don't see kind of a reason you have to restrict yourself to more complete versus less. You can apply this on all of your data. So there is no particular subset of the data that we choose when we are inducing copy masking. Yes.
Probably with that said, it's not complete.
Right. So yeah, the worst case is if something was completely missing in all of your data, then you have no hope of imputing it. But the idea is this would be observed in at least some fraction of your population. And then so let's say it's observed in the first individual, first and the second individual, but missing in the third. And now you might induce missingness into the second individual, right? And try to reconstruct. So this is like a stochastic process where you repeatedly do this and compare your model's predictions to the underlying truth. Yeah, so it's a hyperparameter. So when you're training this model, how much of the data you copy mask is a hyperparameter, and you have to choose it based on like the standard held out or validation metrics.
You mentioned that it could be the method moments could be accurate, the randomized method moments could be accurate because 10 equals 10, which you mentioned was kind of counterintuitive. I agree. So do you have an explanation for why it works with so few 10, and presumably something like the structure of the data wouldn't be super arbitrary?
Yes. Yes. So yeah, it's something we're still working out. I think the rough intuition is the following. So let me give you two examples. An example where we expect it to work, and an example where we don't. So the example where we expect it to work is you have a relatively homogeneous population where everyone has roughly the same kind of relationship to everybody else, right? So what this means is your GRM is going to have entries of roughly the same magnitude, small entries of roughly the same magnitude. And what this means is your trace estimators in the setting, the randomized trace estimators are going to do a fairly good job of approximating the deterministic traces. The setting where it's bad is, imagine you have a couple of people who are closely related and everybody else is distantly related. Okay, so now you'll have a couple of large entries in your GRM and then small entries. And now these randomized trace estimators are going to have much higher variance. And so you'll need many more of them to approximate the deterministic estimators. So essentially, and we are trying to work this out, this has to do with something like the key quantity is kind of the skewness of these entries of this GRM matrix. That's how much intuition I have so far, but it'll be good to understand this more. Right. So depends on your use case, right? So if you're applying this to relatively distantly related individuals, which is what we're doing here, then this works. The skew can also arise due to population structure. So population structure would be a case where you have a few essentially separated entries, right? And then within those, it's well mixed. That's where the design of these random vectors could be useful. For example, you could imagine doing an eigen decomposition, getting the big eigenvalues out, and then applying these randomized method of moments on the rest of this GRM. So yeah, I think definitely we don't have a good handle on the full process, but I think that's the rough intuition that I'm working with.
First, is it some phenotypes follows that are different than normal for either their origin and distribution? And second, when does GCTA, it would try to break down GCTA.
Right, that's a great point. So yeah, MOM, and I just presented normal here because that's kind of an assumption that both that you require for MLE but MOM doesn't require that assumption. So you could, all of this applies for arbitrary distributions with mean 0 and some variance. Right? In terms of the actual phenotypes, yes, the underlying base values of those phenotypes can sometimes be skewed. And this is where how you scale it, what kind of transformations you perform on it before you analyze it can matter. Typically, in these kinds of experiments, most of these phenotypes have gone through some kind of rank-based transformation before analyzing them. So they can be analyzed both by GCTA and by our method. Yeah, we haven't done like a comparison of how different these results will look if you were to apply them to, say, the original phenotypes, but that could be interesting. In simulations, we do see differences where the MLE approaches tend to be more sensitive to deviations from normality compared to the method of moments approaches.